Okay, let's get it started. So if you are in the team, you should be able to see their assignment, homework for you to take a video. Just, I don't know how you can do it. Maybe right now the PowerPoint has the function to let you to present and also at the same time to recording what you're talking about. Just explain to people if you assume people don't know how to do the multiplication of two matrices, and give example matrix A multiplied by matrix B. How are you going to do that using an example, and also explain what that means using some of the language that you can describe. And then after you're done, you can make a small video file and upload to Zoom, and I or Shrine will take a look and see how good you are in terms of understanding. As long as you know what that means, and you get 100% credit. Okay. If you cannot use your language or something conceptually wrong, and then we will reach out to you and explain to you again. Okay. So this week in computer session here, we are going to revisit the slides we talked about in Tuesday, using real examples to practice, to translate basically you learned from the PowerPoint slide into MATLAB environment, and then play with matrix multiplication examples, and confirm what you get is same as what you will do using manual calculation. And also we practice, try to see matrix multiplied by vectors, and the inner product of two vectors, outer product of two vectors. So after go through all these very basic operations, I think we also play with these eigenvalue, eigenvectors. Okay. Then we have a demonstration to use example data. We will download them from a 10x genomics dataset, and see after you download, how are you going to load into MATLAB. And there are other sources which from, for example, geodatabase, from search keywords, and from the reading the paper, get accession numbers. Once you have those, how can you locate the dataset you want to use and pull this dataset out and put into MATLAB? So once you have this capacity, you know how to do it, you can do research and start to follow the papers, using accession numbers to download data, to replace, to reconstruct the results paper authors reported, for example. Okay, so let's visit the slide. As I said, the most important slide, I always have a yellow star on the top. So this is the most important slide in on Tuesday's lecture. We also have another page with a yellow star on it. But now, just using this example to put your matrix A and B into MATLAB, see whether you know how to type that in. And then we start to do the multiplication A times B. Okay? Questions? You have trouble with the login? Okay.
But that one doesn't have MATLAB on it.
Mm-hmm. So this is a small example, only have a couple of numbers. So we just type that into MATLAB using command line window. But I will show you once you have this, how can you save it, save all the variables into a file, and then how to load them back. So you only need to type them once. So what's A? A is contains 6 numbers. So it's 4, 5, 3, and then use a semicolon here to indicate I'm going to type into a new row. Then you have 3, 3, 6. Okay. If you don't want to type this comma in, and it's okay, just leave a space. It should work. Okay, so this will define the first matrix A. It's a 2 by 3 matrix. Okay, you can do the same thing for B. Yeah, I think even before we start to type B in, let's see how we can save A into hard drive. So next time you don't have to load that back to type that again. So if you have a variable, say, already in the memory in MATLAB, you can save this A into a file. You see, save, then give a name of file. So let's give is data A. So this will be the file name. So that then you save A into a file name. So if you do this, you see on the under this folder, you found a new file called data A MAT. So next time you can just load it back. So now if I clear this, I can, so if you clear A0, then you don't have A in your memory, no, not in the worker space, but you can load data file A back, and then you will have this back. So you only write, found a way to make it once, and you can save to the local file here. Okay? Yep.
I think this one also doesn't have MATLAB on it, but do you think that, like, is there a reason why some of those don't turn on? Like, I was, like, messing with this button, but it wasn't working. These ones, I know have MATLAB on it.
But then it's not there
They're
anymore.
not turning on. But
Oh, interesting.
yeah, this one doesn't have that either.
Do you have MATLAB installed here? No, it's not here.
Let me check.
Yeah.
Oh.
Yeah, I hope it's working. Yeah, sometimes this installation doesn't show up because the hard drive has not enough space. It's just,
Okay.
yeah.
Oh, okay.
But normally you should have every machine have already installed.
Okay.
Yeah.
Alright, that makes sense. Thank you.
That's good. So MAT file is a MATLAB data file, so you can save this. If you have a B, you can have both A and a B saved in the same file. So let's see, let's make a B. So B is a slightly larger matrix. So I need to type that in. So B, the rows as 2, 4, 4. Second row, 2, 0, 0. Make sure you have a space, otherwise use comma to separate them. Third row, 2, 4, 2. Then you end the definition of this matrix. You should have a B in the memory. Now I can save both A and B in the output the file. So the name is not good anymore, so maybe I should see data AB. So this will be a new file, will be created in the hard drive here, okay? Make sure you know this workspace is a computer memory. This is a temporary variable is here, but this is a hard drive. It's under the current folder, so you have this saved in your hard drive, so next time you can load it back, even though you shut down the computer, okay? For example, if I clear A and B, so I remove these two variables from the workspace. Now I have no definition of A. I have no variable name in B because they all gone. I wash out this memory, clean the memory, but I can load data AB back. Now they are back, okay? So you have a safe place to save your current variables, your data points, anytime you need. So this is you can also save if you don't want to save into MATLAB data format because you cannot see them unless you have MATLAB. So you cannot open this using, for example, send it to Notepad. It's a binary file, so you don't see it. It's in the data format which you cannot visualize directly. So if you really want to save this into TXT files, for example, you can save write matrix. There's a function called write matrix, then you write this A into a TXT file. So let's see, give a name, data A, TXT. So now you use a function called write matrix. So this matrix can be saved in a TXT file. Then this file you can open using this. Remember, all the files you show the extension. If you don't know how to write files to show extension, Google search that people tell you. So this can be saved in txt file so that you can see using text editor. You can do the same thing for B, and then you have another file. So B. Open outside, so you see your B can be exported into txt file. Txt file you can exchange between different languages. Okay, in R you can read that back into R in Python. So every language have this import. So if someone gave you this txt file, you can also read that in. So let's see. I don't know if we have a read matrix. In this case, will be data A txt. I hope this can read back. So A2. Yeah, so you can save your matrix and also write matrix into text files and also read it back. Okay, so you have the way to save either into MAT file, so matrix, so you have a variable A, you can use save function to save this into a file MAT. You can use write matrix function to save this into txt. So this is MAT, this txt. So this is the function to write into txt. This is save, to save into MAT. I just let you to feel safe. Once you have anything, you have a beautifully computed matrix in the MATLAB, you always have these options, either to save it MATLAB native data file, or you can export, write the matrix into txt, so you can exchange the information across different languages. All right? Yeah. So this assume variable A is a matrix, numeric matrix. It's n by m and fill up with numbers. You can do this. You cannot write two matrices in the same txt file. So you cannot write matrix A and B in the same, but you can write matrix A and B into, save them into MAT files. Because MAT is just like an image memory of your current workspace, okay? Now come back to our definition of A and B. As you know, here A and B has a special meaning. So A, the first row is yourself and your roommate. And the column is the sandwich 1, 2, 3. You want to make 4 type 1 sandwich. And type 1 sandwich, you need bread, butter, peanut butter, whatever. So all the other ingredients is the amount. So now you want to know if you want to do 4, 5, 3, the different sandwiches, how many slices of bread you need to buy, how much peanut butter, jelly you want to buy. So the A and B is a solution to tell you you need to buy 24 slices of bread, and this peanut butter, 16 ounce, this jelly, the other ingredients. But in total, you want to make those 3 types and amount. So that's the meaning of the multiplication of A and B. So after you finish multiplication, you lost information about the 3 types of sandwich. So you only get the total amount of your raw material you want to buy from the grocery store, but you lost the information internally. Okay, the 3 types of sandwiches are so-called latent variables. You don't have those components information. To decompose this, we will have to learn how to do it later, but for now, you just have to know you can compose using A times B, but you cannot do B times A. This will give you the error message because the algorithm doesn't know how you match this internal dimension. That's 3 by 3, 3 to 3. B is so you can do a size of B to get the idea. B is a 3 by 6 matrix. Well, the size A is a 2 by 3, so you cannot use a 3 by 6 matrix to multiply it by 2 with 3 because the inner dimension, they're not matched to each other. But if you do the opposite, A times B, then you have the matched 3, 3, then you can do this, okay? Any questions? Now I want you to separate, to split. Say A have 3 columns. Let's split it into 3 vectors. See, the first vector will be the first column of big A. Let's see, A1 is, so what's the meaning of A1? The first column, that's the number of you and your roommate want to make sandwich 1, the number of sandwich type 1. Yes. So A2 becomes the second type, okay? A3 will be the number of the third type. You want to make 3, your roommate want to 6. So we have this. My question for you is, if you have A1, can you do a multiplication between A1 and B? Is that possible? So what's the dimension of A1 if you do the size A1? The A1 is a column vector. It have two rows, 2 rows and 1, so it's 2 by 1. Let's see whether we can do this. I don't think we can multiplicate A1 by B because B there's no, so there's no one, A1 by B, there's no way you can do it. So A1 to B times, there's no way you can times B. But let's see, we define a vector for yourself. You want to make 3 sandwiches. So the first row will be let's see, B1 is A, the first row. So that's the 3 sandwich you want to make. You want to make 4 type 1, 5 type 2, and 3 type B. And your roommate is B2. Okay, so this time you have the size of your B1. So this is a 1, 2, 3. 1, 3. Now you can have multiplication by B times B. Because B has a dimension of 3, 6. And you have a vector 1, 3. 1, 3, 3, 6. So it match. The end product will be 1, 6. So this is a number you can compute. You can do the same thing for B2. So okay. So just make sure you understand this vector A1, B1, B2 times the matrix B is vector multiplied with a matrix, but it's if you do matrix by matrix multiplication, it's more generalized. If you know you can do that, you know whether you can do this. The vector just one of the dimension in the matrix is 1. It becomes vector, so there's no much difference. Okay, so you can, if you look at the results, okay, this is the same as you do A times B. When you do A times B, you basically put B1, B2 together. This is the same as you multiplied by each row independently. But this is give you the same result when you multiply by when you combine 2 vectors into a matrix. They're the same. You're just using one step to compute. Make sense, I hope. Let's see. We have A here, we have a B here. Let's see, we want to remove. So we want to ignore sandwich type 2. Can we do that? In matrix A, if we ignore sandwich type 2, we can remove the second column. And that in that case, for the B, we remove the second row. Then we can compute. So just ignore the sandwich B, sandwich type 2. Then we have a truncated A. So this will be, let's see, make another name. At first, I copy A into A2. Then I will remove A2 second column into nothing. So this will delete the second column. Now I make a copy of B into a B2. And I can remove Okay, so this will remove the second row of the B2. Now I can multiplicate A2 with B2. I hope you understand what I'm doing here. This has become a still the numbers of you and your roommate, how many slices of bread you have to buy, and the peanut butter and the jelly. It's still the same name, but your shopping list become, the amount becomes smaller because you ignore a set of this sandwich. You don't want to make type 2 at all. So this is just like you have measurement of a cell's gene expression. Okay, it was 24 for the cell for the first gene, but now it's become 14 because that cell shut down one of the gene expression program totally. Maybe it's because of mutation, so you don't have that gene's expression as high as before. It was 24 when you want to make 3 type of sandwiches, but now it's only 14 for you to buy. We will use a lot of those ideas to see whether we can compare this matrix. Think about this as a truncated version or mutated version or knockout version, and then original A by A times Z is a wider type. You see the difference, and then you want to infer whether internally one of the programs have been perturbed or mutated. That's the idea, okay? And sometimes it's important if you believe sandwich 2 is some contamination, then you have a better version of this denoised version of the expression matrix. Okay, you can remove one of the components out of your calculation if you can detect. Okay. Okay, let's see what we need to cover. Do you have any questions? No? I would say this is a little bit abstract. Just play with numbers, but you have to think, okay. Again, for those came a little bit late, we have assignment of homework 1, using your language to explain matrix multiplication, what it is, and how are you going to do it with the example and the meaning of those. Okay, multiplication, when you multiplicate 2 matrices, put it in the context what we discussed here. Take a small video and just show me and Shran, you understand this, okay? We just want to make sure you know nothing else. Okay, I think I need to cover other basics. We need to do so we have a B here. we need to do the transpose. So B, if you put a apostrophe on the top, this will transpose your matrix. Yeah, you can assign this into, say, a B2. Okay, you can do this like B3, and then it's transposed. We can do A3, also A transpose. You can do A, the apostrophe here, and also you can use the transpose. Transpose is also a function, so the same thing, okay? So now you transpose A into A3. Good. So can we do multiplication A3 between A3 and B3? So after you transpose this, A3 become 3 by 2, and B3 becomes 6 by 3, so you cannot make it. But you can do is to put a B3 in the front and then A3 at the end. Okay, what do you get? So now if you look at the inner dimension is matched. B3 is 6 by 3, and A3 is 3 by 2, and the end product becomes 6 by 2. It's just transposed from the A times B. So you can transpose this back afterwards, okay? So this will be almost the same A times B. So make this more generalized if you want to do B transpose A transpose. Okay, I hope you get the idea here. So this function just compare these two items. If they are completely the same numerically, then it gives you the true return is a logical one. Okay, so that means these two matrices are the same. So this is transpose. Now I generate a random number vector and put those numbers into a variable named A. So as I said, most computer science take data points, you describe data points using column vector. So this is a column vector. So basically you define a point with 5 numbers. You can consider this as a data point in the 5D space. You first move to 0.8147 step that long, and then move to the y-axis 0.9, and then move to this axis 0.1, 2, and until you reach to the end of the position. So this is a 5D space point. You can do transpose and then time itself. So this is a inner product. So once you transpose the vector A becomes a vector with a dimension 1 to 5, 1 by 5. And then you multiply it by itself. The dimension is 5, 1. So the end product will be 1, 1. So that's a single number. So that's what it is. So if you transpose A into B, okay, B is the same as A, just different direction. Now you have to do is a B times B parameter. This gives you the inner product, right? Because you change it, vertical A become horizontal. Just make sure you know. But if you make it wrong, so if you do B times B, would this be possible to compute? First, we have to answer this question. So, can this be computed? So what's the B parameter dimension? So this is 1 by 5, 5 by 1. And the B itself is 1 by 5. 5 by 1, 1 by 5. So inner product, the match, it's just very small, it's just 1, okay? So when you do this, you get a 5 by 5 outer product matrix rather than inner product 1. So inner product of two vectors is always a single number, a scalar. But if you do outer product, depends on how long your vector is, it's become n by n, okay? But you can derive everything just as long as you know how to do the multiplication of two matrices. This is just special case to replace the matrix with vectors. And then if you have an inner product, they n by n cancelled out, and you get a single number of inner product. Otherwise, you get outer product, right? Yeah. So if I don't tell you the dimension of B or A, you may get confused. Should I put this times B in order to get the dot product, or for A? So I get confused if I don't tell you. So that's why people develop this function called A dot product. So you always, it doesn't matter, so which direction you gave to this function, it always help you to do the inner product. So this, because we use inner product a lot, more than we will use for outer product. Outer product are also very useful. I will show you a case later. Because we use inner product a lot, so people develop this function just to make sure at the end you always get a single number, no matter what direction either column or row matrix vector you gave to. I don't know if we can do this. Even you put AB, BA, whatever. So dot product is to compute the inner product of two vectors. So if we, by default, we will see a vector is a column vector. So we can do, so this we don't have to do anything for A because it's already column vector. But if you want to make this into column vector, you certainly can transpose it. But also there's another operation is to do this. So this will give you a vectorized version of whatever variable you put in. So basically this just tells you print out all the elements in that vector and using the vertical by default dimension. So this one to guarantee you get column vector. So it doesn't matter is A or B, you get the same thing. So this operation is called vectorization, okay? There's a special name. Just try to reconcile the dimension of the vectors, always put it into a column vector. Okay, we also have this definition of norm, A, okay? So norm should be a single number because the meaning of norm is the magnitude of this vector. So how long from the origin point to this vector. So the distance, basically, that's a single number. So you don't want to use other ways to represent it. So the meaning of this in this 5D, how to compute the norm. So you get the first element and the squared plus the second element. This is by definition, okay? Squared plus the third element squared. A has 5 elements, so we all square them up and then add them up. And then we take a square root. So if you look at the definition of this, it's you get all these coordinate values squared and sum them up and then take square root. That's a norm definition. So this is the same as the norm you compute from this, okay? If it's a 2D, it's easy. So you go x, y, and you know this is a vector length. And the 3D also you can plot this. But when this particular case is 5D, you use the same equation. Just repeat 5 times. And those coordinates can be negative because in this case they're all positive, but if you go negative 0.5 and then negative 0.9, positive 0.1, so you can still move, but your norm will be the same. The distance you move away from the origin will not change wherever you go, because if you have all the same steps, you get the magnitude at the end the same. This is the norm. Any questions for this? We also discussed the unit vector. Unit vector is the norm or magnitude of this vector equal to 1. In this case, this is a small example. A is not a unit vector in terms regarding to this 5D space. It's a little bit longer than the unit vector because the magnitude is 1.65. We can normalize it to let it back to unit sphere in this 5D and allow this to be unit vector. So what you can do is to use A divided by its norm. Okay. In this case, it becomes so let's see, unit. Okay, let's see, underscore u. So this will be another variable. So you basically you normalize the length of your vector and drive it back to unit length. Okay. So this is normalized vector. It's the same data point, but it's just shorter than the origin, the original copy. You bring it back, but the direction of this is the same as the original, small a. Now, if you take a norm function of this, you should get 1, okay? Unified, basically. So if I do norm a underscore u, and this is 1. So those unified or normalized, basically, vectors are useful. It tells you the direction without interfering about any change of the length. You can use this as a definition of your coordinate system because this tells you the direction. For example, if you have a unified vector, so AU is 0, 1, okay? If you're 2D, so people use this as an x-axis standard. And this could be 1, 0, okay? All those are the unified or unitary vectors. You can use this as a coordinate systems to tell you the direction. But this is just more generalized. You can use this as a direction of your x-axis in the 5D space. Tell you the direction. Now my question for you is this A, if I define this as 1, 1, is unified vector? If not, what is corresponding unified vector which is located on the unified circle? Okay. I think I mentioned this in the lecture. How can you compute this? Okay. For the position 1, 1, what's the corresponding unitary vector? So basically the 1, 1 is here. So this is 0, 0. you have 1, 1. This 1, 1 is not on the unitary sphere. So I want to compute what's this point? Okay. This point is the same direction of the 1, 1, but what's the coordinates of this? All right, take a 5 minutes break. Come back. Okay, let's finish off this by looking at this eigenvalue, eigenvector system. Let's first define this A in MATLAB using this very small example here. So I can clean my workspace by clean all, and then do a CLC to clean the command line window. And I define a 2 by 2 matrix A, and I use this as a transformer or operators to do something, to do operations on other points in the space by multiplicate A to that data point. If my data point is the general one in the space, for example, 1, 1. So my data point of V, and remember this is a column vector. Okay, I use A times V, and then the outcome of V becomes 3, 6. It was 1, 1, and it becomes longer. Okay. Oh, by the way, I haven't gave you the answer for this. So basically, this is 1, 1 divided by its norm. Okay. So if you have like V is 1, 1 right here, and then if you do divided by norm V, so this position on the unit circle is this point. Okay. So the norm of 1, 1 was 1.4142. Okay. That's how long this original position is. But after you apply this, and this norm becomes longer, because the operation A makes this data point far away from the origin, make it longer magnitude. And also, if you look at the position, the angle changed from 45 degree to a larger angle. But if this is 1, 2, this is a very special point in the space, then after you do operation using A to transform the position of the data, it only makes the point longer, but not change the direction. There's no angle change. So this data point defines a vector. It's called an eigenvector for A. So if we use function of eigen to find the eigenvector for matrix A, so A has to be squared, okay? It has to be n by n. Then you obtained this vector, okay? But it looks like this is not the same as what we have. We had 1, 2. But this vector and this is opposite, the same direction, just opposite direction, but it's the same vector. So this is different from the 1, 2 we tried. The reason is this is normalized. This 1, 2 bring back to the unit circle. This only tell you the direction of the eigenvector. So if you do if V is 1, 2, if you normalize the vector 1, 2 back to the unit circle, it should give you this number, 0.44. Okay. Basically, all the vectors along this line, this 1, 2 position, this vector direction, all the vectors, they're infinite numbers, depending on how long you think it is. But that direction is important. Define the eigenvector. That's not exactly which point. Okay, so eigenvector is a direction. So in that direction, tell you the transformation of A matrix we've made have nothing to do with this vectors, directions vectors. Only make this vector longer, but not rotated. But any other vectors with away from this eigenvector will be transformed and also change the direction. Okay? I hope you can make a link about this small example 1, 2 as an eigenvector, and then the values you compute from the eigenfunction for the A. Okay? The number you see, the difference is just whether this vector has been normalized and pulled back to the unit circle or not. But that's eigenvector just tell you the direction. That's all we need to know, okay? You may ask, what's a D? D is eigenvalues. Remember, we after transformation, 1, 2 become 5, 10. That's 5 times increase of the magnitude. This gave you the 5. All right. So I said A can tell you, so eigenvalue, eigenvector can describe the characteristics of A. For example, if we do 2 times A eigenvectors, I believe that D should change to 10. So this eigenvalue will tell you the information of your input A matrix. The magnitude contains in the A, right? When you double the A's elements, its eigenvector values also increase. will the direction of eigenvector not changed? The reason is this still the same shape, same symmetric. Even you double every element in there two times, the angle will not change at all. Unless you put one of this into, for example, into 1. Let's try. So A, instead of make this 2.5, make it slightly more dispersed in the position of those. So in this case, I don't think V will change. So you get a different set of the, is it the same? Is it the same vector of the, I don't think so. So in this case, you get a different set of this eigenvector. Okay. And also eigenvalue changed. So this set, eigenvalue, eigenvector, contains information about the structure and also the magnitude of your original squared matrix A. Okay, eigen means in German, it's characteristics or something like that. Okay, the features basically. Okay. So let's go back to what we will discuss about the okay, 10x Genomics, in their website, they have a collection of data set. Using different technologies of their product. So we can select which platform we're interested in single cell creamium. So those are the VISUM and the Xemium, and those are the spatial transcription data. So we are not working on those. So these are the single cell expression data. And then you can also select the universal 3-primer. Flex will have a similar structure of the results. So we can select flex kit as well. 5 primers, so it doesn't matter. So ataxic, those are the epigenetic open chromatin signal. So it's very different from the expression level. We will discuss this integration of different omics data later of those lecture series. But for now, you can select all these standard gene expression matrices we will be able to download from these data sets. Chemical versions doesn't matter. So if you are interested in particular species, cells, nuclear, so this should have other selection operations. And from here, you have human data, mouse, sometimes human cell mixed with mouse. For some internal controls, you can have that. Then the firmware, and we can there are different protocols, prepared samples with fresh samples or fixed samples. Yeah. So I just randomly select one. And then should I have a count? Let me check my count. Yeah, they asked me to put this in all the time. Okay. So inside here, you can have quick view about the report. We discussed the report last time already. And in the output file, we can select this filtered H5. Okay. So this is the file I will download, and then we can load into MATLAB. So back to the MATLAB. The first time loading a little bit slow, but the next time should be okay. Then from here, I select 10x genomics H5 file. And then found it, where I download. Okay, I load this data in. This contains 80,000, 2,000 genes, 12,000 cells. Then we do this using this small filter to do this QC to remove some of the low quality gene and the cell. So this procedure will do an iteration. The first detect the cells which too empty, we remove, and then look at the genes to remove genes, and then we'll revisit the cells, because once you remove the genes, the cutoff, the criteria for the cells also changed. So do the iteration between gene cells until we stopped. There's no cutoff threshold which can remove any gene and the cells, then we end up with the results. So in here, we define either it's more relaxed or more stringent cutoff. More stringent cutoff will remove more cells, more genes, give you cleaner data. But sometimes you don't want to so clean because you want to save most of the cells, even though there are some cells are low quality. You can select either of them. They're generalized, apply to many situations. They are carefully selected cutoffs, including library size, just how many reads per cell. The cell has to contain at least 1,000 reads. Otherwise, there's not much information. And there are percentage of mitochondrial reads that has to be smaller than 15%. Otherwise, it indicates this quality of cell is not good enough. They're most likely to be dead cell, and then mitochondrial membrane is mostly more solid or rigid than cell membrane. So if you have a lot of mitochondrial mRNA released that indicate the whole cell is not in the healthy state. It's most likely to be a died cell. So you want this number to be small. And then you also need to have a minimum non-zero cells per gene. So this is the cutoff for genes. You want to have a gene that expresses at least in 15 cells. If the gene XYZ only expressed in one cell in the whole population, it's very empty. And also in this 38,000 genes, some genes is completely empty. They're all zeros. So we definitely remove those genes. And there are other, one more cutoff. That's the genes per cell. So if I apply this, as I said, the procedure will iterate between cell and the gene until converge stop. And they will report, and I will remove how many genes, how many cells. If you accept the filter, then the data set will be changed to a clean version, and you are ready to save and move to the next step. Any quick questions? We are waiting. We are waiting here. No? Oh, the QC. Yes. So, if you see this small filter icon, if you click that, then you have a selection. By default, the first one. Yeah. So this is a quick report. This many genes will be removed. 260 cells will be removed. So in general, this looks like a high quality data because it's only 200 something cells will be removed. Then we still have quite a lot left, 12,000 and more. And you will end up with 12,256. So this is definitely accepted change. Okay. So those are the cells. Then the second step is to do the embedding. Okay, you select one of the embedding algorithm. You can select multiple. Let's see. I will select 2D this, 3D this, 2D this maybe. So this is just one of the way you can download the data, and then you can do the same thing, download from geodatabase. Let's see, where we are waiting for this. Let's just visit geodatabase to search for some of those. Yeah, I cannot do anything, but you can go ahead and search geodatabase, use some key terms to find the. Not every record in there has a single cell data, because they collect data from the microarray age, bulk RNA-seq, but you have to find a way to search for single cell data in order to use the resource they saved. Yeah, my computer is just locked and freezing there. put all these keywords in, I can be a more targeted search to only return me with the data. In this case, I have 855 results contain these combinations of keywords. And then I certainly will go through some of this and read the introduction of each record to see whether they are the research that I'm interested in to get their data to do the secondary analysis. For instance, I can zoom in this, and they have this keyword single-cell RNA-seq. It seems, yes, this is a single-cell data study, and with stem cell research. But this is not a guarantee you will get good data. You still have to browse down and look at. They do have this 6 samples and labeled with a different experiment from different conditions or samples from different brain parts. And then I have to look at inside to see whether they provide me with the data. In this case, they are not. So it's hard to use. What they provide is combine the 6 samples into called H5AD file. We can discuss this later. This is a Python generated data, and there's a way we can import this, but this data set is too big and 6 samples combined. So I'm not going to demonstrate how to do this, but at least the authors provide me the data we really need. So I'm going to move to another simpler examples. So this is single nuclear attack-seq. This is not what I will use. Single nuclear RNA. Let's take a look at this. Three samples. Looks like exports with NTF alpha at a different time, and then look at this sample. They do have this matrix. Okay. So if you look at the technology they use, it's called a smart seq. So this is not a typical single-cell RNA-seq using 10x genomics. So the data frame structure will be very different. They use the TXT file, basically. It's an Excel spreadsheet like give you the genes, cell labels, and also expression. We can import this into the MATLAB, but this is not a typical protocol and data we want to use. So we can just go through and find until you found the one you really need. I can also start from the papers. Let's see, we work on the Nature Communications paper. We just found an example data, maybe, including single-cell RNA-seq, 10x genomics. If you have a good paper as an example, let me know what the paper is about, and then I can search for it. Otherwise, we have to randomly search as an example. This may be not a good paper because it studied UTR region, and the 10x genomics cannot do any UTR because the barcode, in order to indicate which genes come from, that's not likely to be located in the UTR. So this is not a good paper for us, but I just quickly search whether they have a GEO accession number. So I type GEO, and I will have this access to some of the records. It looks like they use data from other studies, so mouse cell atlas, and in this GEO, also human cell landscape. So they are not the GEO. Another one, they provide some other reference to other large-scale data. They used FLACT sorted, looks like stem cells from here. So maybe there's some examples we can use. So I will try search for this record. So I just need the ID from the paper I extract, and then come back to the place which I can put those IDs in and look at. This is a mouse embryonic fibroblasts. And then they provide CSV file. So again, this is not a very typical, but it is genome 10x genomics. Yeah, so we can download this and also process. So for this example, I can download this file. But you have to know the extension of this file, and also if you know the extension, it's not H5 file. So we have to deal with this in different way, and you have to select the correct options in order to open that file. This file is a CSV file, so there's a way we can do using this to load. But in this case, I can also see, I know the accession number, so I can select accession number and put that number in, let the program to do this automatically. Goes to that record and download this file, and hopefully I can read that in automatically. So if you look at the background output information, the program is visiting this page because you provide the record number, and is reading this file after unzip it. So you don't have to do anything, you just provide this sample accession number, and the program automatically do the things for you. Have those in, and the rest of them will be the same, just filter, then do the embedding. So this is just the whole purpose today is to just let you know the different ways you can get the data in. As long as you know, we can read local files, we can download from geodatabase, we can download using a session number. So different ways, as long as you know how to get into this page, that means you have the data, okay? So that's all we need to know, different ways to grab the data. Okay, what I'm doing here is just download the data and then automatically annotate it. Although I have no idea what their the cell type are in there, but we have a very quick overview of the data points first, okay? So I think that's all for today's computer session, and I will see you next Tuesday in lecture. Remember room 103, okay? If you have any questions, let me know.